Woo, okay, last section, then we're in the clear. Okay, another pivot. We're going to be talking about cells here. So another pop science thing. Some people say that we only use 10% of our brain. Is this true? No, of course, this is completely ridiculous. Definitely not true. For one, if I only use 10% of my brain, then I could have like a couple of strokes and, Phineas Gage's little rod through my brain, and I'd be fine because, it's only a 10% chance of it hitting something useful. That's silly, but it's a little bit underscores how ridiculous this theory is. And I can't believe they made a movie based off of this. But it is true if we think about it from another perspective. The idea that only like 10% of your brain or so is actually neurons. And the other 90% are the accessory glial cells, sort of the behind the scene players that make everything work, right? Like you look at the new Top Gun movie and it's like Tom Cruise and Miles Teller or whatever, and it's but you don't know of the hundreds of PAs and lighting people behind the scenes. So that's the glial cells, and we're going to take a look at them here. So neurons, everybody knows a neuron. We have our soma, our cell body. The axon hillock is the beginning of the axon. So the axon is like the cable that conducts the electrical signal through an action potential. It is wrapped in myelin. That myelin is created by oligodendrocytes in the CNS and Schwann cells in the PNS. There are little areas where it is not myelinated. These are the nodes of Ranvier. The action potentials jump between of nodes of Ranvier, and this quickens the conduction. And at the end, we're going to have these axon terminals, which are going to connect to other axon dendrites. And this is a synapse where neurotransmitters are released. So if we look then at an action potential, we're hovering around -70 as our threshold potential. A little bit of sodium leaks in, not enough. A little bit more sodium leaks in, not enough. A little bit more, not enough. A little bit more, we get to this threshold, this -55. Why is this the threshold? This is the voltage where sodium voltage-gated channels are open. Sodium floods into the cell, this depolarizes the cell around +40. Those voltage-gated sodium channels close, and the voltage-gated potassium channels open. Remember, we have a cell, and it is a salty banana, right? There's sodium on the outside, potassium on the inside. Potassium wants to go out, sodium wants to go in. So now the voltage-gated potassium channel's open. They want to go out. So they start exiting. This depolarizes our cell again. This repolarizes our cell, bringing us down actually past -70 to -80, -90, which is a hyperpolarization. And the sodium-potassium ATPase is transported 3 sodium out for every 2 sodium in. It's going to bring us back to our resting state. Nissl bodies. These are rough endoplasmic reticulum fragments. Remember that the rough ER has a lot of ribosomes. Ribosomes have a lot of RNA. RNA is an acid, ribonucleic acid. Acids love bases, hence they are basophilic. They are going to bind with it's technically actually a positive stain. Too long, didn't read. If you wanted to stain just a cell body and not the axon, you would do a Nissl stain because the Nissl bodies are not in the axons; they're just in the soma area. But additionally, there's more to this. If you get cellular damage, you're going to actually start to lose your Nissl bodies. And again, if the Nissl bodies are staining basophilic, staining blue, then you're going to get a red neuron phenomena. So you get these red neurons is a histological sign that we have ischemia and tissue damage. So a couple markers. So we have in our axons all of these microtubules, but we also have these neurofilaments. These are unique to axons, and so this is going to be a great stain. We also have stabilizing our vesicles with the neurotransmitters. We have a transmembrane protein, synaptophysin. All right, so moving on to astrocytes. Astrocytes, their analog is the satellite cell in the peripheral nervous system. They do like 3 main things. They wrap capillaries with tight junctions and foot processes. This helps maintain the blood-brain barrier. They maintain the neurotransmitter homeostasis, so they're going to provide the building blocks for new neurotransmitters, and they're going to eat up old neurotransmitters. And they are going to be important for a response to injury. Reactive gliosis, which we'll see in a second, friend or foe, it actually might have some downsides. And here's the foot process, and here we can be eating up all of the excess neurotransmitters, providing the building blocks. Here we're wrapping very tightly. I guess that's why I have the bread. I made this PowerPoint a while ago and never finished it. And I guess this is more recycling. Not 100% sure exactly what's the rhyme or reason of all of these pictures, but hopefully you guys get it. How can we stay an astrocyte and not, the rest of the cells? It's because astrocytes have GFAP, glial fibrillary, acidic protein, GFAP. It's only in glial cells. and building. So the point one, you should know that GFAP is in astrocytes. Point two is in response to injury, we have something called reactive gliosis, where glial cells will hypertrophy, they become bigger, and hyperplasia, they're going to divide a lot. And we can see that by notice, we're standing here with GFAP, we see a little bit, and now we see way more after the injury. And this has some benefits and some downsides, and I don't know, I guess we'll say it's more bad than good. I'm not really an expert on it, but we'll say more bad than good. Ependymal cells in the choroid plexus of the ventricles, these are going to generate cerebrospinal fluid, and they have these little cilia so that they can kind of like wave their arms, and you can kind of see them like creating a current, helping sweep the cerebrospinal fluid through the ventricular system. Microglia, they're only micro. It's kind of funny because they're technically macrophages, right? So arose by any other name. Macrophages in bone are osteoclasts. Macrophages in liver are Kupffer cells. Langerhans cells in the skin, right? And so macrophages in the brain are all just microglia. So it's kind of funny that a macrophage is actually a microglia. I guess that's like a little bit of it suggests how large all the other cells are in the brain, because all the other glial cells are macroglia, and so these macrophages are only microglia, sort of as a contrast. If you have HIV, they can become like a harboring site for the virus, and you get these multinucleated giant cells, and they do what macrophages always do: they patrol, they eat up garbage, etc. Oligodendrocytes are going to wrap neurons. So Schwann cells are going to wrap one-to-one. Oligodendrocytes are going to wrap several. So one oligodendrocyte can wrap more than one neuron. You also progressively myelinate more and more axons as you get older and older and older. Multiple sclerosis is going to target these oligodendrocytes and the myelin in the brain. Contrast, we have Schwann cells. It's just like a one-to-one. So here's my Schwann cell, here's my neuron, and it kind of like hugs it really tightly, and then it hugs it tightly more and more and more and more and more and more and more. I thought of like those cute little squirrels from the King Arthur movie. If anything, though, maybe it'd be better to use like the python from like the Mowgli. Rudyard Kipling, Jungle Book. There we go. It's more like the boa constrictor wrapping something up. So you can think of it as a cute, cuddly squirrel. A boa constrictor doesn't matter. It wraps it very, very tightly, one-to-one peripheral. Chromatolysis, really this is chromatolysis and other things. We have three things we're looking at. This is just for axonal injury and neuron injury. So three things will happen. Chromatolysis. So like I think like a chromosome, think DNA, RNA. What are we talking about? We're talking about the Nissl bodies. The Nissl bodies, these rough endoplasmic reticulum fragments that are hanging around the cell are going to start to break down. So the chromosome lysis. And they're going to break down. Potentially, though, if we just say chromatolysis, this is reversible, but up to a point. I think usually it leads to apoptosis. And again, at some point you lose all your Nissl bodies. Nissl bodies have RNA. RNA is basophilic, stains blue. And so if you don't have them, you will have a red neuron. So that's one thing, is we might kind of have our Nissl bodies get broken down over here and then eventually degraded if we go through apoptosis, if it's too severe. The nucleus is going to kind of move over to the periphery. It usually goes even a little bit more over to the periphery. And the cell is going to swell. So a bunch of water is going to come in and cause this whole thing to swell. And then Wallerian degeneration just means that, so let's say that this was where the axon is broken. Everything upstream of this is probably going to be fine, and we're actually just going to build more axon. But everything downstream is going to be basically, this is the Wallerian degeneration is going to be broken down. So this is the macrophages coming in to gobble up all of this extra stuff. Nerves travel in packs. So this whole thing is a nerve, right? So the whole thing is a nerve. And let's start with the axon. Here's the boa constrictor / lovey-dovey squirrel that's wrapping it. This is now wrapped in endoneurium. So the Schwann cell is wrapped in endoneurium. This is our first of three. Now we have a bunch of these axons wrapped in Schwann cells, wrapped in endoneuriums. 1, 2, 3, 4. We have all of these together. All of these together constitutes a fascicle. I think fascism is the idea that like a bundle of rods, each rod together is strong, but like wrapped together, they're even stronger. And that's the idea behind fascism. But you actually have, that's like a structural nomenclature as a fascicle. So here we have a fascicle, a bundle of nerves. and wrapped around this fascicle here is the perineurium. So perineurium is going to be right there. And, now we'll have a little bit of fat, a little bit of vasculature. But again, we have a couple of fascicles together wrapped in what we call a nerve. And on the outside of this nerve is the epi as in outside of the nerve. All right, lots going on here. So we have fast adapting receptors and slow adapting receptors. So fast adapting is a type of receptor that it quickly reaches a homeostasis and becomes uninterested in whatever stimuli it's interacting with. That is useful because if there is the slightest movement, because it's adapted, if something moves, it'll have to that's a new signal for it to pick up on. So it's really good at picking up on the slightest movement. If you're playing the slap game with your sibling, right, where you have your palms facing up and their palms are facing down, and you're trying to slap their hands, you want to have fast adapting receptors that quickly adapt so that you notice even the slightest change. Slow adapting receptors, or if you're holding something and part of your nerves are trying to get a sense of the size and shape and quality and texture, those would be slow adapting receptors. A deep receptor is deep in the skin, and it's going to have a large receptive field. So if you imagine something like your back, right? Think of our homunculus. There's not a lot of nerves dedicated to the back. There's a lot of nerves dedicated to the hand. So let's think about the back. You're not going to have a lot of nerves, so you got to kind of space out the territory. So you're going to have these really deep receptors, which so here's one nerve, and it goes all the way to all of this skin. So in other words, if you poke me right there, which is right there, or you poke me right there, which is right there, it's the same nerve that's picking up on both of them. So in other words, I actually can't differentiate one poke from one area versus a poke in the other area because you've got this one nerve which is overworked and covering way too much territory. So you won't be able to localize touch very well. So here I say more precise, that should be less precise. Then we're going to have superficial, which is, again, look at our homunculus, look at our hand. We've got tons of nerves, so we can have them a little bit closer to the surface so they just cover smaller territory, but that way I can differentiate this poke here from this poke, which is here, because these are both getting picked up by superficial nerves, which again is more precise. All right, let's put all this together. I think of you're going into the alphabet A, B, C, D, E, and not that far into the alphabet, not that deep. In other words, very superficial in the alphabet, we come across M. So M's are the superficial mechanoreceptor. Mechanoreceptor kind of for the dorsal column medial lemniscus. It might be more complicated, but that's how I always thought of it. So the M's are the superficial one, and then you think about, okay, well, who's first in the alphabet? M-E-I or M-E-R? M-E-I, that's first. So that is the fast one, and it is superficial because it's not that deep in the alphabet. This is a superficial fast receptor. These are going to be the fingertips. Now, we also have fingertip receptors that are going to be slow because, again, we might want to detect movement. We also might want to appreciate the shape of something that we're holding. And so this is going to be superficial and slow. So Pacinian and R, these are deeper into the alphabet. So these are going to be deep. So this would be on your back. You're also going to have them in your hands, but let's just to make it easy, think of your back. And then again, we're kind of racing through the alphabet after this tiebreaker. P comes before R, so this is faster. We get to this faster than we got to the R. So this is a fast, deep, and this is a deep, slow. I actually think that's a pretty helpful way to go through it. We then have free nerve endings, which can be myelinated or unmyelinated. These can be superficial here. These can also be deep here. We have photoreceptors for vision, thermoreceptors, which can be free nerve endings. So this is going to be the spinothalamic tract. Chemoreceptors for taste, smell, aortic and carotid receptors for cranial nerves 9 and 10. I think we also have baroreceptors for this, but I could be wrong. Then we have the area postrema, the CTZ, for picking up toxins, for emesis, and then receptors in the hypothalamus, and then we have nociceptors for pain. These are also free nerve endings, spinothalamic tract, and there's actually going to be a bunch of different types of a mismatch of receptors that are all working for pain. So mechanoreceptors is going to kind of be the DCML, and the free nerve endings is going to be the spinothalamic tract. Embryology, there we go. Let's talk about it. So we have this notochord here, which is going to induce via Sonic the Hedgehog factor. So this mesoderm notoderm is going to induce this neural plate to start folding via Sonic the Hedgehog hormone. It starts folding, and we get two different things to emerge from this folding. We get the neural crest cell, which becomes the peripheral nervous system, and then we get the neural tube, which becomes the central nervous system progenitor cells. Note that for the neural crest cells, this is the peripheral nervous system, but it actually covers a couple of things that seem kind of random. Melanocytes, and I always remember this because I remember the substantia nigra. It has neuromelanin. That's why it's substantia nigra, dark substance. It's black. The neuromelanin and melanin and melanocytes are very similar, and so that reminds me that melanocytes are derivatives of the peripheral nervous system. And then the adrenal medulla. Well, we have catecholamines, like serotonin and stuff, and the Rafe nucleus, and a lot of epinephrine and norepinephrine in the sympathetic nervous system. So we also have a bunch of this in the adrenal medulla. So it's making neurotransmitters, which we typically find in the nervous system. So that reminds me as well that this is a peripheral nervous system lineage. And then the microglia. Remember, the microglia are kind of the macrophages of the brain. Macrophages are made in the bone marrow, and bone marrow is mesoderm. That reminds me, this is mesoderm. Technically, though, these come from the yolk sac progenitors, but that's too complicated, so I don't remember it that way. All right, so if we're progressing here, we start with the prosencephalon, the mesencephalon, and the rhomboencephalon, and these are going to become the telencephalon, diencephalon, mesencephalon, medencephalon, and myelencephalon, which becomes, and we kind of went, most of this lectures, we went from the superior to the inferior, the cerebrum, which is the basal ganglia, parts of the limbic system, and the cerebral cortex, the eyes, the thalamus, hypothalamus, midbrain, pons, cerebellum, medulla oblongata, we're going down. Now, how do you remember this? Because that's kind of a lot. I just remember that everything is in alphabetical order except for these two, right? We have M and R. That works. Then we have D, M, MES, MET, and then MYEL. So it's all alphabetical order except for you just got to remember the prosencephalon. I remember maybe like a pro comes first, and then tele is, I don't know, television is on top of your dresser. We're ending with a whimper. That's a bad, dumb thing to say, but yeah. Television on top of your dresser, that'll stick, right? Okay, cool. I think that's the end of everything. Hopefully that was helpful. There's a whole nother lecture, but hopefully I have time to get through tomorrow. Really hope everyone's doing well. Good luck to everybody. All the best. Another one coming.